Thank you, and okay, welcome to the special COVID-19 colloquium. And we've been working on this subject for a while, and before I jump into the details, I want to mention all the collaborators and co-authors we've been very lucky to work with. Most of the work I'm going to be talking today has been done by several students, my co-author, students between myself and Rob Colson, University of Pittsburgh, and here in Toronto, Andrei Vovk and Chad Gu, who have graduated, and now Tiantian Zheng, who should be here, but she isn't for some reason, and in collaboration with the experimental group of Roderick Lim and Larissa Capinos in Basel, Siegfried Musser and Texas A&M, and more recently the group of Edward Lemke in Germany. So just to start as the colloquium talk, just to start from the beginning, and I'm sure most of the people here know about this, but just to get us all on the same page, all cells, all living cells, except for bacteria, which except means there are much more bacteria than any other living cells, but putting bacteria aside, all living cells have a structure which is called nucleus, which sequesters all the DNA, which is the genetic information, and this is fluorescently labeled DNA in the nucleus, which is at this point is spread through the nucleus. It's the blue one, so this is a cell, and this is a nucleus, and this is another cell, and this is a nucleus as well. The reason why, what is it good for, and how did it happen evolutionarily speaking, it's not fully known. There are all kinds of hypotheses, but the lore is that sequestering the DNA in the nucleus enables much higher regulatory and molecular complexity of different molecular species and different molecular networks in the cell. And here I highlighted where the cell is. And roughly speaking, what happens inside the nucleus, there is the DNA, which is in the absence of division, between the divisions. Its main purpose is produce so-called mRNA, which is later translated into proteins. And the access to the DNA for transcription it's initiated by certain molecular factors, and it has to be extremely tightly regulated. So only things which need to go in and when need to go in are let through. And so there is a huge molecular traffic going on through this little hole. There are little holes in the nuclear envelope, which envelops the nucleus and separates it from the rest of the cell. And there are little holes, which will be the subject of the talk, in this nuclear envelope, which maintain all this traffic which goes in, and mostly what goes in are these molecules which regulate the access to the DNA, and what goes out, it's all kinds of things which are involved in later protein production. And this traffic is huge. At every second, in every typical, say, our cell, there will be millions of molecules, millions of proteins going back and forth between the nucleus and the cytoplasm every second. So all this transport is regulated by this molecular machine, which is called Nuclear Pore Complex, and this is, and this is a nucleus of a yeast cell. The green is the nuclear envelope, and these blue guys are these nuclear pore complexes sitting in the in the nuclear envelope and perforating it. But like I said, they're not just big empty holes which let everything pass. There are more sophisticated machines which decide which goes through and in what concentration it's going to be on the other side. And it has several components. So this is an artist's impression of each nuclear pore complex. It's embedded in this nuclear envelope. It's very complex. It consists of proteins of 30 different types. Approximately half of them make this reasonably rigid scaffold, which is very interesting in itself, but it's not going to be the subject of this talk. For instance, it has this famous eightfold symmetry. If you look at from the top, it's composed from 8 spokes. Each spoke's composed of another about 10 or 30 different, 10, 15 different proteins. But the most important thing for us today is this passageway, which is formed by this scaffold of approximately 35 to 50 nanometers in diameter. So for this crowd, roughly speaking, of a large quantum dot. And it's filled with an assembly of very special type of proteins, and they're so-called intrinsically disordered proteins. So you probably all remember from the high school biology that typical proteins, classical proteins, it's a long chain of amino acids which folds in space into some intricate three-dimensional structure, and that's how they function. So there is another very large class of proteins that don't do, they don't fold into any specific three-dimensional structure, and these guys, they're called intrinsically disordered. And these guys which are involved in this nuclear pore complex, they are really disordered. So each one of them can be thought as just long, flexible chain in fluctuating in space. And one has to keep in mind that this is just a cartoon, so this is not a static, this is dynamic structure, which probably changes conformations on the scale of microseconds or so. So what decide, in order to go, so small stuff can go up to, say, 2 or 3 nanometers in size, can go through passively. It doesn't need to be regulated, but anything larger than that has to be bound to special transport proteins. The transport proteins here shown in red, and the black is the cargo, and these transport proteins somehow shuttle things through this whole thing, and that's where the selectivity happens. That's where decision happens, what goes through and what doesn't go through. Now, on the on the macroscopic level of physics, nuclear pore complex is not a passive transporter. It can pump cargoes against their concentration gradient. Obviously, it has to burn energy, non-equilibrium energy, somewhere in the cycle, and I'm not going to go through the details of the cycle because that's not what I'm going to talk about. But it's a very efficient pump. So this shows so this is a nucleus, isolated nuclei of cells, and you can put them in a solution of cargoes, and in the absence of some component of this machine, of this energy burning cycle, they don't go in. Everything basically stays outside. It just binds a little bit to the envelope. Once you switch on, basically once you give them the energy in the form of GTP, for those who know, they suddenly start pumping stuff in the nucleus with very high efficiency. But unlike many other transporters that, again, you might remember from your high school, which work on some very tight lock and key specificity, one thing binds to another, and that's how they recognize each other. It doesn't, because it can't, because the whole assembly, which does the recognition, is soft. It's somehow able to recognize these transport proteins on the background of other proteins on some sort of multiple but weak interactions. And this interaction between the transport proteins and these intrinsically disordered proteins, which I'm going to cover in the next slide, is really the key component of the whole machine. And finally, one final thing I need to mention, that unlike many other machines, it doesn't work one by one. So one transport protein with the cargo count goes through, and then another and another. Simultaneously, there is about 100 of transport proteins inside the hull. Some of them going in, some of them going out, some of the carrying cargo, some are not. So it's also very crowded. And like I said, the key component of this transport is the interaction of these transport proteins with these polymer-like molecules. And I promise this is the only structural biology slide picture I'm going to show. But this is actual structure of these transport proteins, which carries cargo on one side. I'm being a little sloppy, but let's say it's cargo. On the other side, it has these hydrophobic grooves, which are able to bind to patches of hydrophobic patches on these intrinsically disordered, long, flexible molecules. Okay? Any questions about that? Okay. Another interesting thing is that if you look at, say, our nuclear pore complex and yeast nuclear pore complex, and you just naively look at the molecular sequence of the amino acid sequence or DNA sequence or gene sequence of these intrinsically disordered proteins, it's not very well conserved, except for this fact that they have these repeats of hydrophobic patches, which are shown here as FG, after the names of amino acids which are involved. So this red FG is a hydrophobic patches, and that's where I derived their name. They're called FG naps, which I'm going to use in this talk a couple times. And if you look at their sequences, they are not very well aligned. There's quite a very, very variable. But the function is extremely conserved to the extent that one can take our transport proteins, put it in yeast and work, or one can genetically delete some of this guy's yeast and save it with putting in a mammalian molecule, and it works. So which sort of hints, and it turns out to be true, that maybe a lot of how it functions can be understood from basic physical principles, despite the fact that it's an extremely, extremely molecularly complex entity. So like I said, there is lots of different proteins, and each one of these proteins already sufficiently complex, but somehow together it functions the same despite quite large variations in the details. So up until very recently, there was no information, and still it's very limited, what happens inside the hull of the nuclear pore complex, because it's very hard to see what happens in a 50 nanometer hole deep inside the cell. But more and more information comes in, and this is already relatively old data, just to show you what we know and what kind of data we have. So this is a cell nucleus, this circle here, looked from above. Green one is fluorescently labeled transport protein molecule, and these are the snapshots of single molecule tracking, just video microscopy. And you see here it starts outside, it goes into the envelope, which means into one of the nuclear pore complexes, and then it goes in. And this one is the abortive attempt. It starts outside, goes into one of the nuclear pore complexes, and then goes back. So this emphasizes the fact that the process of this transport protein translocation through the pore is decoupled from any kind of energy consumption. It's just essentially it's just thermally activated diffusion. Okay. Second, these are quite old experiments too. In 20 years ago, people were injecting gold nanoparticles into the cells, and this is sort of the sketch of the nuclear pore complex, and looking at the distribution of the nanoparticles inside around that. So this is neutral nanoparticles, pretty large, 30 nanometers, which do not interact anyway but repulsively with these polymer-like molecules, and they're repelled from the pore, while those who can interact, they have been chemically modified to interact with them, they all aggregate where the cloud of these things are. And in the last couple of years, ultra-fast AFM, although it's very somewhat controversial, people still argue exactly what this means, but people are able to come with an AFM tip to a surface of the nucleus, which is covered with these nuclear pore complexes. So for instance, this is one nuclear pore complex, this is another nuclear pore complex, and by scanning they see some sort of dynamics of these intrinsically disordered proteins inside the pore. Another source of information have been in vitro artificial mimics of the NPC, and there were a couple of designs up to now, which all boil down to taking an artificial nanochannel. For instance, one can take a silicon nitride membrane, drill a hole in it with an electron beam, and then chemically attach some of these intrinsically disordered polymer molecules in the hole, and add transport proteins or other proteins or a mixture of those and see what happens. And you can see here what happens on the single molecule level. There are bulk experiments, which I'm going to talk more later. And in the last couple of years, people were coming up with really high-tech mimics based on our DNA origami technology, which allows you to, using DNA, of very special sequence, which makes it assemble in space into structures that can be very well designed. So in this case, it's this cylindrical DNA origami scaffold, which also allows you to put the handles for the attachments of these in very precise fashion. And you can see here, this is the empty DNA origami channel, and this is the one which is filled with electron microscopy, one filled with these polymer-like molecules. Okay. So to mention, well, this is not for physics audience, maybe doesn't care, but NPC, because it's involved in so many processes, it turns out it's perhaps not surprisingly that it's involved in a huge number of diseases. Some viruses need to navigate the nuclear pore complex in order to get their DNA into the into the nucleus, and this is an actual electron microscope photograph. This is the nuclear envelope. These are the nuclear pore complexes, and these balls are what we call capsid, or the particles of hepatitis B virus that carry viral RNA inside, trying to get into the nucleus. It is involved in various neurodegenerative diseases. So, for instance, ALS, that many of you might have heard of. Although it's not known what's the cause or what's the effect here, but these amyloids, which are responsible for ALS, are known to plug nuclear pores. Any questions? So for physicists here, there are two almost separate questions. Because all these artificial channels, they can actually have very direct practical applications. You can use them for protein sorting, for RNA sorting, for viral detection. So the question is, if we want to design a selective nanochannel, which only pass through or detect only certain molecular species and filter everything else, how do we do it? And the second one is specifically pertaining to the nuclear pore complex, putting aside the general principle, how exactly this particular machine works. And for this, we need to understand better the physics or biophysics of this assembly of these polymer-like proteins inside the hull. And it turns out that one can understand a lot about how nuclear pore complex or any similar selective channels work without forgetting even about this about these intrinsically disordered proteins, and thinking about, so because the colloquium, I'm going to talk about some old work as well, thinking about it, translocation of the transport protein is diffusion in some sort of effective potential, which is generated by these polymer-like molecules, which has two components, roughly speaking. One is repulsive component, and there's been a lot of controversy about what is this repulsive component, but one component of it is just the steric repulsion, for entropic steric repulsion from this polymer moving in space. And the second potential one is that they are cross-linked, that they are hydrophobic patches. And the second component of the potential is this attractive interaction between the transport proteins and the polymers. So if the repulsion dominates, we have a barrier. If the attraction dominates, we have an effective potential. And here it's shown as flat, but it's very general. It can be any shape you want. And one can solve this problem and calculate what's going to be the transport efficiency, what's going to be the flux with which you are going through this pore based on the affinity of the particles to the channel. And it turns out, again, this is the thing which can be rediscovered every 20 years. It turns out, well, there's one obvious help from binding, is that it helps you to overcome this entry barrier. But it turns out that the optimal transport is not when it just overcomes it and everything looks flat, but when there is a little bit of potential well inside the pore. And I'm not going to talk exactly why. But these models, they work pretty well. So this is comparison of the theory with the data which has nothing to do molecularly or with the nuclear pore complex, but conceptually it's similar. And it's the work of Charles Martin in the University of Florida, where they were interested in designing DNA sensing channels, and they had these nanochannels, which were, again, without going into details, where they were grafted with one type of DNA construct which interacts with another type of DNA construct, which depends on their sequence, and they could measure the flux of these constants through this channel. So if you have different fluxes for different constructs, you can detect the DNA sequence, roughly speaking. That's the idea. And so I just want to, okay, do I have time? Just one minute quiz. So, well, you see the answer already, so it's going to be an easy quiz. But let's say I have a channel, and I have an empty channel. There's nothing in it. And I'm putting these little wiggly molecules on this side, and I measure the flux through, okay, so it's pure diffusion, so it's proportional to the concentration of the cargo on this side. And now I put these little DNA constructs inside, which bind my red wiggly molecules. So the channel is now stuffed with things, and it also binds my wiggly molecules. Will the flux increase or decrease? You have the answer there. Decrease. Despite the answer, you persist. Okay. Well, that's sort of the most of the people, if they never thought about this, that's what we'll say. Well, it's clogged, so obviously it's going to decrease. Well, it turns out it increases, and that's what this line is. So this is the flux through the empty hole, and this is the flux through the hole with the DNA constants. And it increases because in the absence of these things, the molecule which enters here, because it's just diffusion, it's most likely to go back right away. In order to get through to the other side, it needs to be pulled in, and that's the role of this, and that's what the role of this binding does. And it turns out to be a very general principle. You can increase the transport efficiency by binding, but eventually, if binding is too strong or too many of them, the channel does get jumps, which give you some sort of very general selectivity mechanism. So this is relatively old work, and I basically already explained what's going on, and that's what resulted in this picture, which we call glorified enzyme picture. Enzyme is a catalyst in physics of the nuclear pore complex, where the things which don't need to go through, they experience this energy barrier, but binding to the transport proteins lowers this energy barrier, or free energy barrier really, and catalyzes, if you wish, the translation to the other side. And a lot can be understood within this type of models, and I'm going to return to this in the end if I have time. But it's, of course, but if you want to understand how the nuclear pore complex works, and not just general nanochannels, we need to understand what happens to this assembly of these polymer-like proteins inside. And the problem is that even now, with all the new methods, we still can't measure it directly inside. So for a very long time, a lot of our information came from a lot of in vitro interpretation of in vitro experiments in various settings, which generated a lot of different models with more or less exotic names, and I'm going to cover a couple of them. But even on the level, so let's say 8 years ago, even on the level of in vitro experimental data, there was a lot of controversy. People couldn't even agree if they see the same thing or not, and the interpretation were very different. And one particular setup that was popular is the grafted layers of these polymer-like molecules. You can graft them to the surface, and then you can come and measure the mechanical properties or other properties of this layer with all kinds of techniques, AFM, SPR, and many others. And turns out that if one comes with AFM and pulls one of these chains up, they really behave, if you look at the force extension curve, they really behave like what we know in statistical physics as a worm-like chain model, which is essentially a model of just a flexible polymer. So, which already gives us a hope that they can be thought in the form of some sort of very simplified models. And then you can start adding transport proteins to these layers and see what happens. How does the conformation change? How much you can fit in, and so on and so forth. And so the results of these experiments, they gave an inspiration to so-called brush model, which is named after, well, in polymer physics, grafted layers of polymers are known as brushes. And this model essentially emphasizes this entropic repulsion between the chains and the transport proteins as the main component of the specificity barrier. Another very popular setup was in bulk solutions. And it turns out that some of these intrinsically disordered proteins, of these polymers, some of them that have enough of the hydrophobic self-attractive patches in their sequence, they aggregate, which may be not surprising because if you put enough self-attractive polymer molecules in the solution, they will aggregate and they form this really visible, this is a fluorescently labeled aggregate, which if you torture them enough, can even become almost like rubbery-like. And that gave the rise to so-called gel model, which emphasized this breaking of the cross-links between the polymers as a main component of the specific selectivity barrier. And as we will see, the truth is really in this case, it's really in the middle. But beyond nuclear pore complex, this connects to a very wide world of surface science where people have been working on creating polymer nanoparticle smart materials for all kinds of purposes, for detection, for responsive materials, and so on and so on. So if you think about our FGNAPs as polymers and proteins as nanoparticles, the physics turns out to be is not that dissimilar. So we wanted to know, well, again, this is by now relatively old work. So we wanted to know what's going on. Can we even understand what's going experimentally? Starting from being physicists, starting from the super minimal models, and roughly speaking, the model treats each one of these polymers as a uniform polymer with just some sort of amount of self-attraction. It treats each one of these transport protein as a blob, as a rigid particle of appropriate size, with some sort of attraction to the polymers, plus all kinds of entropic terms. And then this model can be studied in various approximations using mean field theory, self-consistent field theory, simulations, all kinds of things, and they all reasonably agree, which means they're probably absolutely right. No. Okay, so it turns out each one of these chains indeed can be thought as a polymer, as a classical polymer, and for those who know, just using the Flory theory, which says that the size of the polymer is in space. If you take this polymer, it's going to be fluctuating in space, and you can measure the average size using all kinds of methods like small angle X-ray scattering or dynamic light scattering. And the size of that coil, determined by the balance between the entropic terms, we try to pull it and make it bigger, and this internal self-attraction cohesiveness, which tries to make it more compact. And analyzing experimental data using this super classical, super simple model without any bells and whistles at all, it turns out they indeed the behavior is consistent with the predictions of this model, and this internal cohesiveness turns out to be proportional to what you somewhat would expect, to the ratio of the cohesive hydrophobic patches in their sequence to the ratio of the repulsive and charged stretches in their sequence. Okay? Any questions about this? I'm going very fast through everything. Okay. So now what happens if you start... Yeah? Okay, sorry. No. That's an excellent question. So there are two types of. Where is this? So there are two types of interactions. Basically, there are two parameters interact in the model that characterize two types of interaction. One is what you said is the interaction between these transport proteins and the chains, which picture about nanochains be what's inside the channel. And the second one is the self-interaction between these polymers. It comes from the fact that they have these hydrophobic patches on them. The main purpose of these hydrophobic patches is to bind the transport proteins, but because they're hydrophobic, they're going to self-associate as well, and some people thought that it's very, very important. So it turns out that at least the observed behavior can be really well described. So we really can think about them as simple polymers, at least up to some limit. So now what happens if you start adding the transport proteins to those kind of assemblies, not individual polymers, but to the assemblies of these polymers? And in the beginning, there is this repulsive pores, or repulsive interaction, which tries to exclude them from the assembly, which can be, like I said, overcompensated by the binding of and surface interactions of these transport proteins with the polymer. And the free energy of these interactions can be calculated, and indeed, if you increase the interaction strength between them, eventually it becomes negative, indicating the penetration into the layer. And there is one interesting point, physical point, is that this cost of the entropic cost of inserting a particle into assembly of these polymers is not additive with the energy gain which comes from the interactions, because they can wrap around it in some really non-trivial ways, affecting the entropy of the whole thing. So that's something you need to be kept in mind, that at some point things become less simple. So this is a simple picture. If they're interacting enough, they're going in. If they're not interacting, they're not going in. But if they're interacting enough, they start penetrating the layer, and this is the result of the simulations. The red are the backbones of these chains. The blue are the particles. But then at some point, they penetrate the layer to sufficient density so they start filling each other. And some sort of microphase separation occurs where they start forming a dense layer, dense and compact layer, tightly intercalated particles with the polymers near the surface with some other stratified dilute thing, until eventually everything is compact, and if you add more stuff, you can only swell back. And you can do it for different particle sizes. The effect is a little bit different, but it persists. So this is important because it might tell us what is exactly the dynamic, conformational dynamics, or the spatial organization of this assembly of these FGNAPs and transport proteins in the pore. And it turns out that this is what happens experimentally. And I have to say, so not to brag, but we did not develop the theory to fit the data. We developed it completely in parallel, and it turned to work. We had to play a little bit with parameters to make it work with the data, but qualitatively it's, and this is the experimental data for those grafted layers, which I showed before. And as you add more transport proteins, the height of this layer goes down, and then it starts to swell more. And this is exactly what happens, and the theory even reproduces the difference between the large transport proteins and small transport proteins and things like that. Moreover, it turns out it's a more general phenomenon known in polymer physics as Coenen solvency, which is a very interesting thing where you have, let's say, a polymer layer of grafted polymers, and you put it in one solution, and it stays swollen. It doesn't collapse. You put it in another solution, it stays swollen, it doesn't collapse, but then you mix these solutions in some fraction, suddenly it does collapse. And that has to do with the fact that one of these solvents interacts attractively with the polymers the same way as our transport proteins interact with them, with the chains. So that's the second piece of evidence that our simple-minded polymer models works. This is the third piece of evidence. So we were encouraged, and we went and applied it to these bulk aggregates, which are formed if you put enough of these polymer-like proteins in solution. And this is And like I said, it stands to reason that if you put self-associating molecules in solution, they will aggregate, okay? And sometimes it's going to go through some sort of phase separation mechanism. And again, this is described extremely simple model known as Flory-Huggins theory, which includes some minimal entropy terms plus these self-cohesive interactions between the chains. And it does make predictions for at what concentration and what interaction strengths they're going to phase separate. And the red and black line are the predictions of the theory. And the dots are the experimental data from a variety of different, from the same type of this molecule, but from different species. So they're all a little bit different, but more or less the same. And there's only one indeterminate parameter in this theory, and that's this effective self-association interaction, which we can determine from here. Okay. So now, what happened? Fortunately for us, a group of so this experiment were done by Dirk Gerlich, who was one of the biggest proponents of the gel idea in Germany, who are extremely meticulous experimentalists, and they took these aggregates and they checked how permeable they are to the transport proteins, to check the idea that those kind of aggregates have selectivity, so that some things don't go in, some things go in. And what they saw, if they put transport proteins, these two, they go in. So this green indicates going into this assembly. So this droplet is this assembly in space. These guys are fluorescently labeled. You add them, they go in, it goes blue. But now you start adding a larger and larger cargoes on them, and they stop going in. But even the very large cargo will go in if you slap enough of the transport proteins on them. And if we want to be consistent in our physical description of this, we should think about it as a three-component solution now of polymers, transport proteins, well, and a solvent, but solvent is always explicit, and see what's the right thermodynamics of these kind of mixed systems. And it turns out, and it's quite interesting, and I'm going to, and it exhibits phase separation in various places, but I'm going to focus on this phase separation region. So if you increase at some concentrations of transport proteins and of these and the polymers, the phase separation occurs, and if either the interaction strength between the two is high enough, or the size is small enough, then the concentration of the transport proteins in the dense phase is higher than the outside, which indicates permeability. But if you say increase the size of the particle, the whole phase diagram, well, it undergoes some sort of fusion, and then these tie lines flip, and the concentration of the transport proteins in the dense phase is lower, indicating no penetration. And if you now go and plug all the numbers, and again, there's only two indeterminate parameters in the model, and one we have already determined from the analysis of the phase separation without the transport proteins. And now if you plug everything in, it turns out that the agreement with the experimental data is very good. So these relatively small transport proteins, they penetrate very well, and then the larger ones do not. And but even the larger ones will go in if you put enough transport proteins on them. Okay? So I'm going to skip quite a few slides. So to summarize, what is the simple physics we learned from all this? So at least on some level of coarse graining, these intrinsically disordered proteins, which have in principle very complex molecular sequence, they can be thought as simple polymer chains, self-associating polymer chains with self-association roughly proportional to the balance between the hydrophobic and non-hydrophobic residues in their sequence. At high enough concentrations, they will phase separate, and the permeability of these bulk assemblies is both, as well as the permeability of simpler grafted assembly surfaces, is determined by just a balance of two factors. One, there is a cost of inserting a transport proteins in, which comes from entropic repulsion of because of thermal motion of the chain, potentially breaking of the cross-linking, osmotic pressure. Osmotic pressure can be different in a denser assembly. That's the cost. And the gain is just to the attractive interactions between the particles and the chains. And that seems to, at least to the degree of accuracy of the experimental data, this is enough to understand what's going on, at least in simplified mimics of the NPC. Any questions about that? Okay, so in the remaining 10 minutes, I'm going to talk a little more about more current work. So the main question is, so this is all nice, but the problem is that the nuclear pore complex, like I said, it doesn't work one by one. It's not like one transport protein goes through, goes through. It's extremely crowded by transport proteins carrying cargoes in both directions. So just to be a little cute, it doesn't look like this, but more or less look like this, or maybe even like that. And the question is, how does it maintain this bidirectional transport? I mean, everybody who's taken the TTC subway in rush hour knows this problem. So the question is, how does it optimize the bidirectional transport without clogging? And there's been a number of ideas, so there is some potential evidence, although it's also controversial, like everything in this field, that potentially there might be some pathway separation. So some types of cargoes, shown here in green, they take, let's say, more peripheral pathways through the channel, and some other takes more central pathway through the channel, and that's how they don't block each other. But before going into that, but the second related, but not exactly the same problem, which we addressed before this one, was what about non-specific interactions? So there is tons of stuff in the cell that can interact weakly but non-specifically with the pore, and there is so much more of it in the cell because the cell is a soup, so there is so much more of other stuff in the cell compared to the transport proteins. So how come it just doesn't clog it all the time? How do transport proteins are able to carry their function on the background of all this non-specific noise? And that can be also investigated with using those very simple models, extending them now into two species. You have a channel, there are two species of particles. You can use some hopping model or diffusion models, which takes into account interaction between the particles. It takes into account the crowding, so that if there is a particle here, the next particle cannot move. In physics, they are known as exclusion process theory. And something very interesting happens. It turns out that if I take two particles, which somewhat interact with the pore, and I take one of them and check how fast it goes through the pore, it goes well. I take the other one, it goes well. But when I mix them, the one which goes just a little bit better completely exclude everyone else. And there are even some weirder effects, which I'm not going to go into the excess of time. So there is some really kinetic mechanism built into just this very simple design. And for some people it's obvious. Some people, because those which go through better because they bind stronger. So of course they occupy more space, so they exclude everybody else. But it's not that simple. It's really a kinetic mechanism, because one could imagine what could one imagine? One could imagine two scenarios. So one could imagine, for instance, that it will enhance the transport non-specific one, because they can get trapped between the two specific ones and go through better. But in reality, what happens is this. So that's what could happen. That's what happens if you have a strong competitor. If you have two particles which have reasonably similar binding to the pore. Now, if you have one particle which is strongly interacting with the pore and therefore goes better because it penetrates better, and you mix it with the other one, which doesn't interact as strongly, what happens is mostly this. So once it, sorry, my bad. So once it comes, it has to wait because the pore is blocked by the better one. So it most likely goes back, and that's why it filters out much more efficiently than actually you would expect from thermodynamic considerations. So why do these strong ones don't block themselves if they're spending so much time in the pore? And the reason is that if this one comes, it can wait until the other one passes through, and then he passes through itself. It takes longer, but they pass through in higher numbers. And these ideas have been tested in one of the earliest nanochannel mimics by my long-term collaborator, Tijana Jovanović-Talisman, where she managed to take nanochannels, graft some of these polymer-like molecules inside, and then test the transport of different transport proteins in mixtures or non-mixtures. And that's exactly what they saw. They saw that, well, it's just names. They saw that the transport protein goes through with some rate, which is similar to the transport rate of a control protein, which has same size and charge, but doesn't have specific interaction with the pore. But when you mix them together, the transport of this non-specific guy, which in our case models on this non-specific competition in the cell, it goes down significantly. So just in the last 2 or 3 minutes, so now we are in a position to actually say something about the actual pore in a little bit more realistic scenarios and start answering these questions about pathway segregation and the transport of actual biologically interesting object through the pore, which is just to show you, to give you the flavor. This is the setup of a simulation of the nuclear pore complex. The red ones are these chains, and the blue and yellow ones are the transport proteins which are going in or going out. This is another example which we are now doing in collaboration with Edward Lemke in Germany, who have developed a battery of modified viruses which of different sizes where they can also control the degree of their interaction with the pore, which enables us to test really in really precise fashion how does the interaction and how does the size affect the transport properties, and what do we need to modify in these theories. So just to summarize, these extremely minimal, extremely coarse-grained physicist models of this very complex biological molecules, they seem to work pretty well. At some point, they start to break down if we want to know more details about what happens, and that's where we are at. And so we are now, like I said, we are poised to start hopefully answering some actual real biological questions, and also to start to combine our now understanding of this structural part and the kinetic part of the transport with this active cycle around it, which makes it into a pump, which makes it into a machine through energy consumption. And maybe it's on the level of speculation, there is always a discussion if biological systems, if they are fine-tuned or robust, in a sense, if you encounter a very complex biological entity, whether every single detail matters for its function, or you can sort of tweak them and untweak them and still function functionally in the same way. And nuclear pore complex is an interesting example to think about these things, to see what is general and what is very system specific. So I think I'm done. Thank you very much.